So much, everyone, for coming to join me on the journey of looking at milk cells, which is the study of my, is the topic of my research. So today I'm going to be talking about marvelous milk cells and how this is going to help us unravel the secrets of the human breast. So just a few housekeeping notes. Please put your phone in silent if that's possible. There's no fire alarms today. So if an alarm sounds, then I think we've got to get out of here. We're broadcasting at the moment. And so this session is going to be recorded and will be available online after the event. So for the online audience out there, welcome. Please submit your questions during the webinar using the Q&A button and they'll be addressed to me. And we'll do the best that we can to address any questions. However, I have put up my Twitter and my there's like a little QR code on my slides if you want to contact me directly to ask some questions. Fantastic. So why study human milk cells? Well, this journey of mine started when I was an undergraduate and I was looking to start a research project. And at the time, I went to a lecture as part of my degree, which was talking about cells in human milk. And I thought that was really fascinating. At the time, my mum was breastfeeding my little baby sister, and I thought, wow, there's cells in there. What are they there for? What are they doing? And what can we learn from them? And that's where I started my journey doing a PhD, looking at the milk cells and now postdoc. So this is one of my favorite images of a breastfeeding mum and baby. And what we can think about when it comes to this relationship is there's not just the mother and the infant, there's also the milk there. So there's this mother-infant triad. And basically, each of the members of this relationship have the ability to impact upon the others. So looking at the composition of human milk, well, it's mostly made up of water, as you can imagine, which is there to hydrate the baby. But there's other components in there as well. So we have different minerals, there's proteins, lipids, and carbohydrates. And what's really fascinating is they actually have the ability to interact with each other as well as with the baby and impact the infant's growth outcomes. So what we have are oligosaccharides. These are human milk oligosaccharides, and these are basically carbohydrates that feed the microbiome of the infant. There's also milk fat globules, and so these can obviously provide some nutritional impact for the infant, but are thought to also help in other areas as well. We also have antibodies, many different kinds of proteins, as well as the bacteria, which kind of infiltrate the infant's gut and help them to grow, as well as maternal cells. And of course, the maternal cells are the things that I'm really interested in for my research. So from the maternal perspective, how the mum can impact the milk composition through her diet. So depending on whether she has a fatty rich diet, different components can actually go in from the bloodstream to the milk for the infant, but some are actually synthesized by the breast, and we're still trying to untangle exactly which components are coming from the diet versus from being synthesized within the breast. Also, environment has an ability to impact the composition of the milk as well. And from the infant can impact the composition of the milk based on the age. So basically the lactation stage. So how old it is, this changes the protein content of the milk and different other factors of the milk as well. Now, obviously, as I mentioned, this milk has the ability to profoundly impact on the infant. So there's a number of different health outcomes that are actually benefited by the process of human lactation and by breastfeeding. So there's a protection against mortality and morbidities, such as necrotizing enterocolitis, which is a condition which unfortunately affects many premature infants. And this is where there's an infection within the gut. So human milk's been shown to help support that and make sure that that doesn't happen. There's also protection against diarrhea and respiratory infections. There's also a reduction in the risk of overweight and obesity, as well as type 2 diabetes. There's some evidence to suggest increased intelligence. Of course, it's a nutritional supply, but there's also immune protection, and this assists in development. And I should also say here that, of course, I'm a huge breast milk fan, but I think that feeding the baby in any way that's possible, I totally support. So a fed baby is a happy baby. So one factor, of course, that's coming from the mother that can impact the composition of the milk is making sure that the mum has an adequate supply. And unfortunately, this isn't always the case. So there's some women who aren't able to breastfeed. And I really want to find tools to help support them. That's kind of one of the reasons why I'm doing this research. Obviously, also the milk composition can also impact the infant. So as I mentioned, there's these oligosaccharides, these complex carbohydrates that are actually in the milk, and these feed the infant microbiome. And then that kind of helps determine the health status of the baby as well, making sure that they're nice and healthy. So from the maternal perspective, there's also many benefits that the mother receives from breastfeeding her infant. Of course, there's an increased attachment to the baby, but there's other factors that can come into play as well. So from the maternal perspective, there's a reduced risk of obesity as well as breast cancer. And this relationship of breastfeeding and breast cancer is a really fascinating one. So there's an age-dependent decreased risk in developing breast cancer following childbirth. And if you can see on this plot here, so if you look at the blue line, this is for women who've had children when they're less than 25 years old. And initially the risk factor, the risk ratio for developing breast cancer slightly goes up, but then long-term it goes down. And as I said, this is age-dependent. One wonderful thing is that for women that breastfeed, every 12 months of breastfeeding decreases the risk of developing breast cancer by 7%. However, in some cases, if someone does develop breast cancer during pregnancy or the first year postpartum, there's an increased metastases and mortality. And so for me, I'm really interested in trying to understand how does this process of pregnancy and lactation actually influence the composition of the breast, and how does that basically lead to these decreased risk of breast cancer as well? So yeah, this is a big question that still remains in the field. So I want to talk you through the changes and the stages of what's happening in the adult human breast. So in its resting state, I'm going to use this word resting quite a few times. This basically means it's not doing anything. So not pregnant, not lactating breast, not making milk, which is obviously the whole purpose of the breast is to make milk for the baby. So in its resting state, we have this ductal tree, and it's embedded in stroma, so lots of fatty tissue there to make sure it's well supported. But it only really kicks into gear when during pregnancy in a process called secretory differentiation. So here we have a lot of the ducts growing, we have a lot of the functional units of the breast starting to grow. And so this is now the breast is primed to make milk, but it's not quite ready to do so. It does so after the delivery of the placenta, when there's a drop in progesterone, and we get a process called secretory activation. And so at this point, this is when you get a little bit of colostrum, and then after that, you get full milk production after this. Now, after this process of lactation, so when the infant is weaning and there's no longer milk needed, the breast then can return to a somewhat resting state in a process called involution. Now, I say somewhat resting because there's actually evidence to suggest that there is a fundamental change that happens to the breast after this process. So it's not exactly the same as it was before. Some animal studies suggest there's an epigenetic memory, as well as a potential arisal of new cell types as well within the breast. And so how this impacts the long-term breast cancer risk, we're still not sure, but there is some evidence to suggest that if someone has a second child, that their milk will come in a lot faster. It's almost like the breast knows what to do. So what I'm really interested in is to understand what's driving these changes on a cellular level, on a molecular level, and I'll do my best to bring you along for the journey on this and explain basically how I'm studying this. So looking a little bit more in detail about the breast. As I said, what we have here is we have kind of these grape-like structures on the end of these straws, I guess, which is the straws of the ducts that lead the milk from the functional unit, these grape-like structures, to the nipple to be delivered to the infant. And within these grape-like structures, we have two major cell types. So we have luminal cells. What we see in the middle of this kind of grape-like structure is a lumen. Hopefully that will help you remember it. But basically the luminal cells are the ones that are producing the milk. Around the outside of these lumens, we have myepithelial cells or basal cells. And these ones actually squeeze the lumen so that the milk can then go through the ducts and then be delivered to the infant. And as I mentioned, surrounding these grape-like structures in the stroma, we have a lot of stromal tissue. So we have fibroblasts, we have fatty tissue, and this kind of helps support and keep everything in place, as well as the vasculature as well. So we've got blood vessels and things like that to make sure that there's plenty of blood pumped through the breast. So what you've got here is a FACS plot, a fluorescence activated cell sorting plot, and I'll explain a bit more about how this process actually happens. But what you've got to realize here is that I've got two plots two plots, and basically each dot on here represents a single cell. And along the first plot, this forward scatter, that's helping us understand the size of the cell. So how long it is along the x-axis is how big it is. And along the y-axis, we have this CD45 and CD31. So these are two proteins on the outside of cells that we can use to help us figure out what kind of cell they are. So in this case, CD45 is an immune cell marker. So this is a blood cell marker. And CD31 is an endothelial cell. So this is a vasculature, one that helps make up the blood vessels. And so actually what we're interested in the mammary gland field is mostly the epithelial compartment. So the compartment that's actually functional during these changes that happen during pregnancy and lactation, as well as during breast cancer. So if we look at the cells that aren't immune and endothelial cells, that they're actually epithelial cells, we can use this marker EpCAM, which is along the y-axis, and CD49F, which is along the x-axis. And we can separate out the different cell types that I've kind of highlighted to you already. So what we have is we've got these 4 clusters. One of them is hormone responsive cells. So these are the cells that express estrogen and progesterone, and they're the ones that respond to the hormones. We have luminal progenitor cells, and I'll tell you a little bit more about them in a moment. And we have these basal myepithelial cells, and these are the ones, as I said, that contract to help the milk being delivered to the infant. Now, the luminal progenitor cells are quite interesting. For the lab that I work in particularly, because they're thought to be the cell of origin of basal-like breast cancer, so a particularly aggressive form of breast cancer. And what's really fascinating is some animal studies have suggested that these luminal progenitor cells are actually the cells that differentiate to become secretory during lactation. So this is really interesting to understand the dynamic of how these cells are changing. So obviously, when we're trying to study the cells within the breast during these very key stages of development, we don't want to be going and asking women to donate bits of their tissue. So we do this in a slightly different way. We ask some women who have undergone a breast reduction surgery whether they'd be happy to provide the breast tissue that they're already discarding. And we can use that obviously to study the different cell types and the different effects of whether they've had children or not on the breast itself. So these are called mammoplasties. And what I've also discovered is that we can isolate cells out of human milk to then actually be able to study them. So how we do this is I have obtained some breast tissue from women, again, who were happy to donate because they were having a breast reduction surgery. And then I chopped it up, added some enzymes, and what you get out is single cells. Now, alternatively, from human milk, what we can do is actually just centrifuge it. So spin it really, really fast, and then the cell pellet comes to the bottom of the tube, and then we can use it to study the lactating breast. So here's a picture of me looking at some cells under the microscope. And I want to show you what they look like when we look down the microscope. So I definitely fell in love with milk cells when I started looking under the microscope. As you can see, it's a bit of a milk mess. And basically what we have here is we have some cells in here, but we also have milk fat globules. And so milk fat globules are different kind of circular membrane enclosed structures that contain fat. They're delivered to the infant, and we're still kind of uncovering the functions of them. Now, looking at this picture here, when I was a young undergraduate student, I thought, yeah, of course I can see the cells, you know, there they are. But actually, we have to use a nuclear stain to help us identify which of these things are actually cells. So you can see the things that are green. This is the nucleus. So this is where all the DNA is contained. And so not everything that looks like a cell actually is a cell within the milk. Some things are actually just milk fat globules, which are the kind of the units that are expressed into the milk. And so this is a close-up of a Marvellous milk cell. So what we see here is we've got the nucleus, as I said, containing the DNA, and we can actually see the lipid droplets in there as well, so the milk fat. Now, I'm such a big fan of milk cells that I've kind of dressed as one today. I don't know if you noticed. I've got my nucleus here and I've got my milk fat right up here as well. Yeah, so I think these are a fantastic way to kind of study how what's happening during lactation, because these cells that are in milk are actually actively producing milk. So that's something that's quite unique. So just thinking back to what's in a cell, I'll give you a quick little overview because it's going to be useful for the following slides. As I mentioned, we have this green stained nucleus containing the DNA. The DNA is then translated into RNA, and this is kind of, we can use this as a gene expression tool. And then we have protein. So you might want to think about each cell has hundreds of different pieces of RNA, hundreds of different, thousands, I should say, different types of protein. And basically, we can look at these to characterize different cell types to really understand what function they're performing within our body. So if you think about the DNA as a library, the RNA is like a recipe and the protein is the product of this whole process. Now, if you look at a cell and you think, okay, it's making these different things, it can help us classify what kind of cell it is. So in this case, it's like a milkshake bar, basically. That's the cells in human milk. So I say, what kinds of cells are there in human milk? It does seem to be clear that there are some cells that are producing milk, but looking at the literature, there seems to be many different kinds of cells. And so I really wanted to dig into this further to definitively say what kind of cells there are in human milk. So according to the literature, there are many different kinds of cells in human milk. We have epithelial cells, we have immune cells, mesenchymal cells, secretory cells, progenitor cells, and endothelial cells. And these are all using 1 to 2 markers to characterize these different cell types. So as I mentioned, when we're looking at the cells in milk, or any cell really in the body, it's going to have DNA, it's going to have RNA, and it's going to have protein. And focusing specifically on the RNA and the protein, these are the things that we can use to classify the cells, to determine what the function of the cell is. Now, the RNA is encoding for a specific kind of gene. And so this gene is basically then going to be transcribed into protein. And we can look at the RNA using sequencing and the protein using flow cytometry. And I'll talk you through these processes as I go. So fluorescence activated cell sorting. What we're doing here is we're taking a mix of cell types, as you would in any part of the body if you want to extract some cells. And we're looking at the proteins on the outside of the cell. So the proteins on the outside of the cell are going to help us understand what kind of cell we're looking at, because it's giving us an indication as to the function of the cell. So what we can do is we can put this mix of cells into a machine, a fax machine, where they're passed through a laser beam. And each of the cells have got kind of an antibody on the outside. So if it expresses a protein, if it has a protein outside, it will be recognized by the laser beam and it will be captured by the filter. So then we can understand what cell type it is, whether it's expressing a particular protein or not. And then we can sort them based on that. So we can better understand kind of the function of these cells based on the proteins on the outside of them. So looking at cells from breast tissue, this is very similar to the plot that I showed you before. So I've done this myself and I've used the CD45, which is this immune cell marker. I found that there's some cells within the milk that are expressing this. There's a large proportion that aren't, and the ones that aren't can then be separated into these 4 populations. Now, obviously, I was quite curious to do this with the cells in milk to see whether they had a similar profile or not. And actually, surprisingly, they don't. So we can't use the markers that we would use for normal breast tissue to help classify them. There's some fundamental differences, the cells in milk compared to the cells in the breast. So we do have immune cells as far as we can see from this plot here, but we don't have these 4 perfect populations of breast tissue cell types within the milk. So human milk cells have a different profile to cells from the non-lactating tissue. So One thing I also did kind of around the same time that I was realizing that they don't have the same profile was I wanted to grow them in culture. So what this is you take the cells that I've taken some breast tissue cells, I've cultured them in the petri dish, kind of the classic, science thing that you would expect to see. And basically what I've seen is that these cells can grow. So this is the breast tissue cells. So each of these little mounds is a single cell, and the bigger mounds are made up of multiple cell types here. This is from the breast tissue. So this is well established, nothing too groundbreaking here. I've also found you can do this with the cells in milk. So the cells can actually grow, and we can use this to better study them. Now, another model that's been developed is to put breast tissue cells in a matrix. So what this structure that you're seeing here, this is derived from a single cell, and it's growing ducts and it's growing alveoli so that we can really understand in a 3D way how the breast is looking. This is derived from breast tissue. What I've found though is that we can actually also do this from cells in milk. So we can generate a 3D model to start to study the process of human lactation. And that's something that I'm hoping to take forward in my future research to build a model in the lab to then be able to understand how milk is actually being produced. So live tissue and milk cells can be cultured in 2D and in 3D conditions. But again, we're still thinking back to what are the cell types in human milk and how do they differ to those in the resting breast tissue? So to do this, I wanted to use an unbiased approach. So I didn't want to use all of the markers or the proteins or genes that have been described in the past for the cells in human milk. I wanted to try and figure out a way to do this without putting too much bias on it. So again, I took some breast tissue from women who donated it after a breast reduction surgery, and also some cells in milk from women who were breastfeeding and were happy for me to do these studies. I recruited 7 breast tissue donors and 9 milk donors. And I should say, being involved in milk collections is such a wonderful experience. You have such a wonderful community of people who are so interested in learning more about human lactation. So I was really grateful to everyone who provided samples for this. So from these donors, I took the samples, either the breast tissue or the milk, and I prepared them as I showed you before. And then basically these cells were all in like little Eppendorf tubes, and we can load them onto a machine. So this machine, the cells are in this yellowish color. So each of these little yellow cells will be encapsulated with a bead. And what this does is in this little droplet, we have a single cell, we have a bead, and this will then barcode each of the individual pieces of the RNA. And what this means is that we can understand for each cell what the gene expression profile looks like. So we can help to classify the different cells within either the milk or the breast tissue. And from this process, we sequenced quite a lot of cells. So it was about 100,000 cells to really give us a great understanding as to the cell types within the breast, as well as the cell types within the milk. So this is a lot of information. So we have, in our genome, we have over 20,000 genes, right? So there's over 20,000 genes that could be expressed by a single cell, and we have about 100,000 cells. It's a lot of information. So the data sets that I'm working with are quite huge, but to try and organize this data into a way that makes sense is I wanted to see whether cells were similar or different. And if they're similar or different, then what are the characteristics of those cells? What's the function of these cells? So I kind of want you to imagine a map. So if you think about a map, if we've got people scattered across the globe, people who are closer together might have more similarities, and people that are further away might have more differences. Not always the case, of course, but this is just kind of an analogy. So hopefully that sets the scene. What I've done with the cells is I've also mapped them, where I've taken the breast tissue and the milk samples, and I've mapped them. So each of the dots, tiny, tiny dots, we've got 100,000 cells on here. Each of these dots is a single cell. And if they're similar to each other, they have a similar gene expression profile, they're going to be close to each other. And if they're pretty different, they'll be further apart. And so each of these dots is colored by which sample or which donor has provided it. So the pink cells are coming from the milk, the blue cells are coming from the breast tissue. And what you can see here is that we've got 6 kind of clumps of breast tissue dots. And we've got two major clumps coming from the milk cells, and we've got kind of a mix up there. So don't worry, I'm going to tell you what cell types there are. So to do that, what we've done is basically, I've just got the reference of the breast there, just so I can help refer back to that. What I've done is this is the same map that I've just put up here. And what this is showing us is whether a particular gene is being expressed. So this gene is PTPRC, which encodes for CD45. So if there's a high expression of this particular gene, it's going to be in red or yellow. And if there's pretty much no expression, it's going to be in blue. And hopefully what you can see from this image is there's a kind of a mess of cells that are sitting up the top there, which maps to the cells that come from both the breast tissue and the milk. And so I've classified these cells based on many markers, not just this marker, as immune cells. So the immune cells are coming from both the breast tissue as well as from the milk as well. I've done this for each of the clusters, so I'll try and go through them fairly quickly, but P63 is a very well-known transcription factor. It's expressed by these basal myepithelial cells, the contractile cells within the breast, and I'm only seeing them from the breast tissue, so not from the milk. PCAM1 is a marker that encodes endothelial cells. So these are the cells that help make the vasculature, make up the blood vessels. Decorin is a marker for fibroblasts. And finally, we have an endothelial cell marker. And so what you can see is the endothelial cells, the fibroblasts are coming from, oh, sorry, one of those meant to be vascular accessory. All of those are coming from the breast tissue alone. And what we're left with in the middle is two clusters from the milk and two clusters from the breast tissue. So I found that they both express, or they all express keratin 18, which is a luminal cell marker. And so looking a little bit more into detail as to what these different luminal cells from the breast are, I've termed them luminal progenitor cells, which hopefully might sound like a familiar cell type, as well as hormone responsive cells. So that's progesterone receptor there. But we still have these two unknown clusters that are quite different from the cells within the breast tissue that we have to really understand. And as I said, they are luminal cells, but we want to characterize these much better. So from what we can tell, milks containing two luminal epithelial cell populations. I've termed them LC1 and LC2 for luminal cluster 1 and luminal cluster 2 because they're a bit unknown and I have to classify them further, and the immune cells as well. So this is again the same map. I've just colored it slightly differently again, just based on the cell type now, not based on the sample that they're coming from. And what I've done here is I want to understand the differences between LC1 and LC2. So what I've done is I've used the gene expression profile. So as I said, each of the cells that I'm looking at has thousands of genes. And I want to see what's similar or different between them. So what you're looking at here is a volcano plot where each of the dots is a single gene, not a single cell this time. And so what we have is some genes are significantly higher within the luminal cluster 2, and some are higher within the luminal cluster 1. The purple ones are the LC1 and the pink ones are the LC2. And so this gave me a bit of a headache, I'll be honest, because it's a lot of genes. I didn't know exactly what they were doing. I thought, how can I figure this out? And so what I did is I've used some software to help map genes that have been shown to be performing particular functions when they're working together. And so I've used a database, which is called a gene ontology, and that's to help me figure out what these different genes are actually doing. So from the luminal cluster 1, what I found is that they have a high level of transcription. So they're actively turning the DNA into messages that can be read, the RNA. Whereas in the LC2, what we're seeing is higher levels of oligosaccharides being produced, as well as many other metabolic processes. And what's really interesting about this is it's suggesting that the LC1 and LC2 cells might actually be producing different components within the milk, which I think is really fascinating. So LC1 and LC2 seem to have different functions in milk production, and we're still kind of unraveling these. A lot of the studies that have been done on lactation haven't been done in humans for obvious reasons, and so there's still a lot of work to be done to uncover exactly the role of this LC1 and LC2. So another question that I had is, do the cells in milk actually interact with each other? So we have these luminal cells, there's LC1 and LC2 in milk, and we also have a range of different immune cells. And it's really interesting to think, are they doing something? Are they chatting to each other while they're actually in the milk? So each cell can basically speak to other cells using a molecule called a ligand. And if you think about that as spoken words, like I'm speaking to you right now, and cells also have receptors on the outside of them. So these receptors bind to the ligands. And if you think about the receptors as being your ear, so I'm communicating a message to you that you're understanding. I don't know if it will make you go off and do anything differently, but in the case of the human body, that actually is what happens. So if cells will communicate to each other, that usually means that downstream there's a process that happens as part of that. So I wanted to see, as I said, whether these cells were talking to each other. And this is based on their gene expression profile. So it's not definitive proof, but it's giving us an indication as to whether the different cell types are expressing certain ligands or receptors, and whether there's a possibility that they're interacting. So this little plot here is to basically help us understand whether there is communication happening and how much communication. So all of the cell types are represented around the outside. And if we're seeing a strong signal, so a lot of communication happening between one cell type or another, one of these, the lines will be thicker. Whereas if there's not so much communication, the lines will be thinner. And hopefully from looking at this, you can see that actually the LC1 cells seem to be really actively communicating with the different immune cells. So digging into this a little bit further, I've looked at the pathways of which genes are being, which signals are actually happening between the different cell types. So along the bottom of the circle is whether the cell is basically expressing a ligand, so trying to communicate, and along the top is whether it has a receptor, and it's colored by cell type. So you see a lot of purple. As I said, the LC1 cells seem to be really loving communicating between the different cell types. What we have first up is epithelial growth factor. So what we're seeing here is that the immune cells seem to be communicating to LC1, and epithelial growth factor obviously helps the cells grow. So maybe that's helping the cells grow. Midkinin is a hormone. SPP1 is osteopontin, which is really important during lactation. And we also have a range of different pathways that are associated with immune cells. And so for the MHC class 2, what we can see is that the LC2 and LC1 cells seem to be really actively communicating with the other immune cell subtypes. So we've started to identify that there might be some signaling that's happening within the immune cells and the luminal cells. What that means in terms of how it impacts the baby's immune system, how it impacts milk production, I'm still figuring that out, but I was pretty excited to see that there does seem to be some sort of communication that's happening between these cell types. And this is kind of really helpful because in the breast, the epithelial cells and the immune cells communicating with each other can change basically what they're doing. So this has been shown in breast cancer to be really important, but it obviously might be really important during the normal function of human lactation. So the final question that I will try and answer for you today, based on the data that I have, is which cells in the breast are most like milk cells? And obviously this is important to help us understand how the cells in the breast have actually changed to then be able to produce milk during lactation. So here again, we've got this map of the cells. Hopefully it's becoming a bit more familiar to you. And what I've done is I've tried to basically look at the characteristics of the cells within breast, so which genes are being expressed by these breast cells, and see whether these characteristics are expressed by the LC1 and LC2 cells from our milk. So what I've done is I've made a signature score. And so what this means, this score is hundreds of genes that are being expressed. And what you need to know is along the y-axis, if a cell type is similar to the cell type on the score, then it's going to be higher up. If it's different, it's going to be lower. And so what you should see from this plot here is the cells that are labeled HR, hormone responsive, have a very high level of the hormone responsive score. So that was very good to see that the genes that I was using were very accurate. If we think about our basal or myepithelial cells, this is the basal or myepithelial score. And again, they're highly expressed by the basal cells, but not so much by the other cell types. And finally, looking at the luminal progenitor cells, we can see that the luminal progenitor cells from the study are highly expressed, but so are the LC1 and LC2 cells. And so what we're getting from this is that the milk cells seem to have a similar profile to the luminal progenitor cells. So milk cells most resemble luminal progenitor cells from the breast. And so using this information, I wanted to understand, how did the cells in the breast, these luminal progenitor cells in the breast, actually transform during pregnancy and lactation to allow it to produce milk? And so what I did is I compared LC1 and LC2 with the luminal progenitor cells from the breast. And again, thousands of genes differentially expressed. So I had to try and figure out what they might be doing. And there's still some work to be done behind this, but using the publicly available data sets, what I've done is I've looked and I've seen that for the luminal progenitor cells, these cells seem to be performing different functions to the cells within the milk. So they seem to have some function associated with maternal processes, which is not that surprising. There's an upregulation of cell adhesion markers, so how much the cells stick to each other. And that's also not surprising because obviously we've taken these cells directly from the breast, whereas the cells from milk have been floating in milk. And so they wouldn't be having these adhesion markers. Now, really interestingly, looking at the purple plot, so looking at the gene ontology terms that are associated with lactation, so the milk cells, lactation is one of the top ones, unsurprisingly, but we also have many other biosynthesis pathways, including lipid synthesis and different metal ion transport and things like this. And so this is giving us a really good hint as to the different processes that are occurring during lactation. But as I said, there's still tons of work to be done to really understand this. And that's something that I'm trying to do moving forward. So milk luminal cells most resemble luminal progenitors, but clearly have different functions. And obviously milk production is one of them. So to give you a little overview of what I've shown you today, is that we can study cells in milk to help us better understand the status of the breast during lactation. As I said, there's still much work to be done, particularly in understanding how different metabolic disorders might affect this. Human milk contains two secretory luminal clusters that are different from the non-lactating cells, but they do resemble luminal progenitor cells. There seems to be an enrichment of certain pathways associated with milk production, such as lipid production in the secretory milk cells, and there's a downregulation of cell adhesion pathways. And what we think is that this work provides a rich resource for constructing and exploring biosynthesis pathways associated with milk component production and transport. So I really want to take these studies further. And so I've got many, many directions I want to take this research, one of which is I actually started milking mice, which might be a bit strange, but in that way we can actually look at the cells from the milk in mice, and we can look at the breast tissue from the mice and compare those directly. So firstly, I really want to understand is, how is each of the thousands of different components of milk actually produced? And that's something that still needs to be looked into further. And how is this affected by maternal genetics? Why can't some women produce enough milk? Is there a reason? Is there a signaling reason? Is there a hormone reason? That's something that I'd really like to uncover with my research moving forward. And finally, can we look at cells in the breast milk as like a way to help us understand whether breast cancer might happen in the future? Because obviously these cells are coming directly from the breast, they're epithelial, and I think that they could be really useful to giving us a non-invasive way to look at what's happening in the mammary gland. And so with that, I'd just like to thank you guys for being here. I really appreciate it. It's great to see so much smiling faces, and also to present to an actual audience. It's been a while. So my work is embedded within the CALID research group, and we've got a Twitter handle there. It was started in Germany in the SHIEL research group. I have many different collaborators that have helped me with this project, many different funders. The QR code you see there is for my research page. And also I've just started the Cambridge Lactation Network as well, to try and bring together different members of the research community to help us understand some of these really complex questions around human lactation. So thank you very much for your attention. We got any questions from the audience?
Yeah. Got an online audience.
Sorry.
Okay, never done a mic before. You said you had 9 mothers providing you milk samples. Were they all at the same stage of breastfeeding? Because you said the composition changes.
Yes.
So would that have impacted on your research if you had them at babies are 2 weeks old or babies are 2 years old?
Yeah, I think that's honestly a fantastic question. So when I initially started this study, I tried to recruit quite closely. So the women were at around 2 months of age. However, after reviewers' comments, we had to recruit a lot more. And so that's where I had a few more that were a bit later stage of lactation, as you say. But there's actually been an excellent study that's been published looking at the cells in human milk using the same techniques that I have, and have found differences in composition as the baby's aging, which I think is absolutely fascinating. So it's a really great question. And yeah, I think we can try and understand maybe what's regulating the differences in milk composition by looking at these different cell types across these stages. So good question. Yeah.
That enormous reduction in diabetes that you mentioned at the beginning for people who've been breastfed, is that screened to be entirely about breastfeeding, or are there other factors maybe in there too?
Yeah, I think that's a fantastic question. To be honest, I don't know the answer to that, but I do think that it's worth looking into. So these are from epidemiology studies, and so the mechanisms behind these reductions in risk aren't actually known, and that's why I'm really keen to understand these changes that happen in the breast. But yeah, wonderful question as well.
I'm really interested in what you were talking about in terms of whether some women can or can't produce enough milk. Have you done any, are you looking into any of the sort of situations that those mothers might be in? So whether they're stressed, for example, or in abusive relationships, or?
Yeah, I think as you say, okay.
I was really just asking how much is stress a factor in, and how does, what actually happens when a woman is stressed and her milk dries up?
Yeah, I think that's a wonderful question. As you say, there's so many different factors around making sure that a woman feels supported to make sure that she can actually breastfeed. There has been a few studies looking at the cortisol levels and how that might actually impact milk production. I'm not sure exactly of the mechanisms behind that, but I think that moving forward, definitely in my studies, I'm going to start to consider these questions as many factors as possible to understand why some women can't produce enough milk. So whether, as you say, it's stress related, which I think is totally valid and could definitely be an issue. I remember I was collecting milk from someone and they were so stressed out, and they just kept saying the pump's not working, it's not happening. And I said, you just have to try and relax. And then suddenly the milk came out with no problems. So yeah, I think it's a huge factor. I'm not exactly sure of the mechanisms right now, but I think it's definitely worth studying further. Absolutely.
We know that cow milk protein in the membrane causes clotting. Well, at least in vitro, it causes platelet aggregation. It affects NK cells, natural killer cells, and therefore maybe predisposes to cancer. Whereas human milk doesn't do that. So is there any research going on about the differences between human milk, especially the fat globule membrane, and the cow milk, well, and goats and sheep and all the rest of it? Is any of that going on? Because roughly 40% of our population die of heart disease. People with advanced heart disease have high levels of antibodies to milk fat globule membrane. So it seems to be an important thing for someone to look at.
Yeah, absolutely. There's actually quite a lot of studies now going on the milk fat globule membrane from humans. So kind of historically, this field of lactation was mostly in the dairy field. So people weren't really interested as to how these different effects were actually affecting humans, which is quite surprising. So there is research ongoing. I think it's absolutely fascinating. And I think as well, within these milk fat globules, obviously they have a membrane and different proteins on the outside, but they also contain some of the different components of milk producing cells as well. So there's also RNA, there's microRNA, there's different organelles. And so I think that there should be done a lot more research on this, which I'm happy to see actually a lot more people are starting to study human lactation. So yeah.
Thank you for a very interesting and clear lecture. This may sound like a stupid question, but do you think that a woman would lactate indefinitely if a child carried on suckling?
Ooh, that's a really good question. I mean, I can only speak from my experience, which is collecting milk from many, many different women. And I have seen some women breastfeed for quite a while, actually, like to 5 years old. But whether it would happen indefinitely, I actually have no idea, to be honest. It's a great question. And what impact that would have on the breast, I'm not sure, but I think she'd be quite exhausted, to be honest, because there's so much energy that's taken up by milk production. So I wonder if there might be some sort of an evolutionary conservative thing to try and protect the mother as well, so she doesn't keep on going. But I think in most cases, as far as I understand, it's usually a rather active process that a woman gives up breastfeeding. However, there is some evidence to suggest that if she undergoes pregnancy, that the milk composition actually changes, and that means it's less palatable to the infant, and so they actually stop breastfeeding. So yeah.
Thank you. Why is early pregnancy, I mean the pregnancy in the early 20s, protective? And is it the pregnancy or is it the lactation that's the protective factor? Whereas in subsequent pregnancies, or in a woman that perhaps has a baby in her 30s, 40s, I think it can be the opposite. It can actually increase the risk of cancer.
Yeah, I mean, so basically these studies have been epidemiology studies. So they've been looking at thousands of different women and just correlating, how these risk factors are actually happening. But we don't actually know the cellular mechanisms, and that's something that I'm really interested in understanding. So yeah, it's a great question. It's something I really want to answer with the research, is trying to figure out these age differences, whether there's different cell types, whether there's yeah, exactly. Cell types, different signaling. I'm not sure, but I think that's something that we're really interested in understanding more. Fantastic. Well, thank you all for being here. That's been really wonderful seeing you, and I appreciate showcasing my research. So I think there's one more slide here. Let's have a look. Yeah, thanks so much. So if you're interested in revisiting this, it will be available on the YouTube channel or the festival's SoundCloud. So thanks again for joining me today. And if you've got any more questions, please feel free to get in contact. So thanks a lot. I appreciate it.